So I still have the statistical digital twin on my screen, and we can see the grains in red and the binder in green, and actually I should label those by going to the view and in the legends and overlay select the material legend to be on. So the air or pores are invisible here in white, and the solids should be grains, and then the green is the binder. To compare this twin with the real data set, we should actually run the chord length distribution and identify grains on this statistical digital twin. But we actually happen to know that the porosity is much higher than for the real data set, because I interrupted the structure generation a moment ago. The porosity is actually way too high. We still have about 5% more overlap than we should have, and that that overlap would actually reduce the porosity. So instead, and because the chord length distribution couldn't even be done with the 2021, let's pretend we had run those two analyses, and let's compare the data that you downloaded instead. So I'll go to the file section here, and we'll open results, but not the results that we have computed. Instead, I go to the building statistical twin folder where you have the results that were pre-computed, and actually pick the two chord length distribution, the chord length GDR and the chord length validation GDR files. And they're loaded into the GDR viewer, and actually I will pick both of them. So highlight by hitting Control left mouse button. Both of them are highlighted in blue right now. And I will move this piece here over to the left, so they look actually very much the same, and we can see that the mean chord lengths agree very much between 9.39 and 9.41 in the x direction. In the z direction though, for the original data set, the chord length was 8.6, and for the validation data set's chord length was 9.1. So it's a bit longer in the validation data, and so on. So what one can then do is actually combine the results by selecting, pressing that button up here, and I will import both of those, overwrite previous result as I have done it before, and then in here I will plot the results of both of those analyses, so you can compare in a single graph the data of two different analyses on two different structures in the way that is just shown here with combined results. I'll actually show you that one can focus on that picture by changing the GUI a little bit, and we see that the results are rather similar, except there are shorter chord lengths for the real data than there are for the validation data. So there's one more thing I'd like to point out to you. There's also a way to look at Excel by creating Excel single table, which is different from the export down here. The window just disappeared on me, so I'll go back up here, and here's all the data that we had earlier exported to Excel for your further analysis if you need it to happen. And I'll go back to the original GUI again. Now let us compare the output of the grain identification on the original data and on the statistical digital twin. Again, we use the pre-computed values from the hard disk. So I load results again. So open results up here. Have to go back to the data that you downloaded in the building statistical digital twin folder, and this time I choose the identify grains and identify grains validation GDR files and open them. I highlight again both of those, the last two that I loaded, the identify grains and the grains validation, and make them equal size over here so that I can compare them, and I see that the grain volume percentage is close for the initial as well as the after the boundary grain removal. The grain volume statistics is close, and the grains vericity statistics are very similar, grain diameter statistics are similar, and the coordination numbers are let's say okay. The tensor is a little bit off in here. Now I have all these data that I don't want to compare just by looking at the numbers, and instead I'm going to actually combine the results again like we did for the chord length distribution. So I combine them, and I keep the default name here, except I have already used combined GDR, so I'll call that combined identify grains GDR up here, and say OK. So now I have the two in one, and I can actually now look at the graphs that compare both of those. So let's look at the plots. Let's look at the scatter plot at first and go through that in some detail. I'll look at them in the maximum way, and first I notice that the color bar is actually on different axes, so I have to unify those, and I'll go into the first one, the upper plot in here, and I can select a lot of options in here. So I'll go into edit plot section, plot settings, and I will not have an automatic color chosen here. Instead I choose the color axis range myself. I'll change that value to 2 over here, and apply that, and I'll do the same thing on the second graph down here. Oops. Go in here and edit the plot settings and disable the automatic color scale. Choose the maximum value 2 here as well, and apply and close, and have to close this again here. Now the two histograms can be, the two plots can be compared directly with each other, and the similarity in the colors really means the similarity of the actual structures. So this is not the only quantity to compare to make sure that we really have a statistical twin in here. So we'll actually look at all the other options here as well. So for example, the threshold volumes, there is actually a distribution of those, and in this plot we see the identify grains and identify grains validation, that is those of the statistical digital twin, plotted next to each other both as curves and histograms, and they are very, very similar, as you can see. Next I compare the volume histograms, and again both of them are very close. The purple color is where both of them agree, and in one case the identify grains validation, the statistical twin, is a bit higher, and in another case the identify grains or the original data set is a little bit higher, but they agree very much. Next we look at the vericities, and here we see that actually the original data have a slight tendency to smaller values, and the statistical digital twin has a slight tendency to somewhat larger values. But yeah, that's the best we can do so far. Let's look at the diameters also for of the spheres. So you can look at not just diameter of volume equivalent spheres, but also at the shortest fit diameters, intermediate fit diameters, and longest fit diameters, and for all of them the histograms look very, very much the same. Next we compare the coordination numbers, and in here we again see a somewhat bigger difference than in some of the other plots, where the statistical digital twin is a little bit more to the right, and the original data set is a little bit more to the left. The red data is slightly left of the blue data. Next, we compare surfaces that are very close to each other. We can compare contacts that are very close to each other, surface smoothness, which agrees, and surface to volume ratios that you can compare in here. And last but not least, we're going to look at the orientation. So each and every one of the fitted ellipsoids has a long direction, and that long direction points a certain way, and we will compare the two between these two polar angle plots. So first of all, I want to take the legend out of this, so I go into the plot settings in here and remove the legend up here, and then I don't want the color bar, the color axis to scale automatically. Instead I'll set it to 0.25 to be able to compare both of those plots. And I'll go into the other plot as well, edit plot settings. Don't want to draw the legend, don't want to scale the colors automatically, and choose the same maximum value for the axis range here as well. And now one can really compare the two plots right here, and one can see, even for the orientation, which is I think the least good match, a fairly good agreement between the statistical digital twin and the original data set. So why is it interesting to have a statistical digital twin? Well, one can now do statistical digital siblings and look at what if scenarios to find better performing materials in the computer. For example, let us remove the binder, and I go to Model Process Geo, a very powerful module that we haven't used at all so far, and in there I will reassign a certain material ID, namely everything that was binder before in green, and I will make that air again, so porous. And I reassign that, and all the binder is gone. I move back into Grain Geo. I still have the add binder functionality open here, and instead of adding 3% binder, I'll actually add 6% binder right now. We've done it before. Actually, I'll call it statistical twin binder 6%. Say OK and generate that. And now GeoDict is adding twice the amount of binder to that structure compared to what we had done before. Actually, by using the binder identification functionality, one can even do this for scans of real materials. Remove the binder and add different amounts and varying number of other parameters to it. So let's take a look what the structure looks like right now, and we see that the pore space has reduced in volume, and there now quite a lot more binder is visible in the image here. I can slice through there to give you some impression of that. So there are all these other aspects of the work that we have done that I will now get into. I'd like to review our work on the statistical digital twin in a few different ways. Now, first, let's look at the hard drive. So I go to the user data folder that we created in the beginning of this session, and I have it sorted by date modified, and we see the statistical twin with the binder with 6% binder in it is the latest addition to this folder, but we can see also the combined identify grains GDR, the combined GDR for the chord length distribution, the statistical twin with binder original, and the first statistical twin that didn't have binder in it, where we actually created only 50% solids instead of 53%. There was a chord length distribution run in here. We saved a 2D image, and in the identify grains we also saved an MPEG file, an animation of just 2 of the grains that were analyzed. So GeoDict does not crash very often, but even if it did, all results are saved to the disk, so nothing is lost really. So this is true not just in terms of results, but this is also true in terms of all your operations. So there's another very important folder besides this one, which was our project folder for this particular session. So all the data that that had to do with digital material design ended up in here. There's another very important folder, and that is the settings folder, and I have prepared that one as well. So it's on the C drive on Windows machines and Linux. There's a similar location. Please look at the manual for that information. And in this settings folder, so it's in users Wiegmann GeoDict 2021. There are 2 particular folders that I will have a look at with you. One, there's a log folder, so it shows that I have a log file still open just now. That's the current session of GeoDict 2021. But there are also older log files, and GeoDict usually keeps log files back for about 2 weeks, but we see that I have less than that because the beta is not even 2 weeks old actually in this case. And inside those files, let's take a look at them. We find when this particular session with GeoDict was started a few hours ago, and then everything that happened and that was written out, which you didn't see, all the things that were done by you, in particular all the input parameters, they actually end up inside this log file. And if there is something going wrong, this file is what you can communicate to us, and we will be able to find issues if there are any bugs, or if we can support you with doing something better. That log file is a very important step to do that work. There's another folder in here, and we will get to that in the GUI as well, which is called the session macro folder. So I go in there, and you again see a number of files that date back to the beginning of when the beta was released, and they're actually all Python files. And I'll open one of those for you, the most recent one, the most current one, and I'll actually go to the very end of it, and we'll see what happened in here. Actually, I see here an add binder arcs command, and then at the very bottom there is a run grain geo add binder. So that was the 6% binder that we added, and then actually GeoDict all by itself, it loaded also the result file. So the statistical twin binder 6% GDR was loaded into the GUI and displayed to me. So those were the last 2 operations we actually did in GeoDict, and they are available in this Python file. So how does that look in the GUI? And yeah, please note that this makes everything you have done completely reproducible. Yeah, you have all the parameters available to you, and you can take portions of this file and recombine it and have your workflows done in Python, and have reproducible research that you can share with your colleagues, that you can run again a year later and to get the same results as before. So in the GUI, there is in this macro section here, there's also an entry session macro, and I just showed you the location where this sits on the hard drive. And in here, also in the GUI, you have accessible all the operations that you did since you started this particular session of GeoDict. We can actually select all the commands and add all the selected commands to a file and save that file. And I'll do it just for a demo right here. I'll call that session.py. And so you don't need to look at the session macro folder in the way that I did earlier, but you can also save all the commands or some particular ones. And I'll look in the user data folder. Now here's the session Python macro. And in here is again basically the same content as we saw earlier. I like the folding of all the information. There's a little bit of documentation in the file, and then after that you see this long list of things that happened. There's a pattern here. There's arguments, and then there's an operation. You know, there's an identify grains right here. There's a load GDR files. There's a save result plots, run some built-in macros, etc. So the everything that you do in GeoDict can be expressed as a command, and the command usually has the syntax of GeoDict load GDR file or grain geocreate, etc. That makes sense when you look at the GUI. And so that by getting to know a little bit of the syntax, you can get familiar with how the how to automate and how to program GeoDict yourself, and that will be the topic of the 4th session tomorrow afternoon, actually. So as this meeting is on image-based simulation, I will actually go back and use the last few minutes of this first session to show a little bit of work that one can do with the original images. So I go back to import GeoVol and browse back to the original cathode data. So into the downloaded folder, and there I'm going into the grayscale folder again, and I again pick the first TIFF image from the stack in here. And this time I won't crop on input, but I will load the whole 1400 by 1400 by 500 data set. Again, we import all files in folder, and I will set this to 500 nanometers, 0.5 micron again, and go into the image processing section. So the data I have in memory don't fit with this new data set, and so it asked me if I want to discard that in this case, and I'll actually answer yes. So I have enough memory also to look at everything in this case. And we now see the complete data set that we had cropped to 500 by 500 in the X and Y direction has now the full length of actually being 1400 by 1400 in the two length directions. And before that I went rather quickly through a lot of the visualization options in here, and I will now spend a little bit of time on that. So I actually go to the full size of the images, and I will switch off the segmentation again, and for example I might want to see the XY view alone and complete, and I should go to the Z direction in here, and then we can see that this is now the original size of the data set that one can run analysis on, of course, as well, but everything would take significantly longer, hours instead of minutes in this case. And we see all the different ways of visualizing. Of course, there's the X and Y direction that we had seen earlier, the histogram I talked about earlier. There's also a 3D preview, and if we go back to the tile view, then we see that 3D preview is actually changing depending on where I am at with the 2D sliders right here. And there's a couple more pretty interesting features, namely the gray value analysis for the X, Y, and Z direction. And this is an important quantity sometimes to do corrections by actually reducing artifacts from 3D scans to have that red line, the quadratic fit of the corrected data set, actually be a horizontal line, indicating that the same gray value means the same thing in the image after applying a correction based on these curves right here. So I'll go back to the stacked view here and look at just the XY data set. Maybe I'll look at the complete image, but remember it's shrunk down by a factor 2 already right here, and show a few more of the capabilities of the image import right there. So for example, we could crop the image not just on import, but by inspecting it first. It would take more memory because we would have to load the whole image, and I could say I want to see what the cropping would do, and actually slide the screen line in here tells me where I am cropping actually in the X direction and maybe also in the Y direction. So there's now the upper right rectangle would be left from these crop operations. Doesn't make any sense right now. I don't want to do it, so I undo that. There's even a suggestion for an automatic crop for you, for example. There are other options, like for example embedding into empty space in any of the 3 directions, or this one could be interesting. GeoDict is able to recompute everything to a different resolution. For example, sometimes in FibSEM images you have a different dimension in the Z direction. For further analysis, maybe you need everything resampled to a uniform grid. That can be done here. Where actually with size FibSEM, GeoDict will recognize their realized distance in Z and do non-uniform sampling even on loading the data sets for FibSEM values. Or let's look at downsample. So this is just by a simple factor 2 changing the size, which you could also achieve with the scale here, but with a simpler user interface. So there's all kinds of things about the image size, the image alignment, that sometimes the 3D scan would need to be rotated, sheared, straightened, or permuted. There are gray value adjustments, resample to different bit rates, enhance contrast, correct flickering, correct gradient brightness, Gaussian brightness, invert the image. All kinds of image processing operations that for this example we're fortunately not needing them, but they are available in the software. The image filters you have seen, and I really try to encourage you to play with GeoDict a little bit yourself, maybe with this data set or with your own data sets while you have the evaluation license for the next 3 weeks. And that's it for the first session, and I thank you all for attending, and I look forward to your questions.